This protocol allows researchers to generate pseudotyped viruses that can be safely used to study viral entry events of highly pathogenic viruses, such as SARS coronavirus and MERS coronavirus. This versatile technique is based on the easy setup transient transfection. This system can also be applied to produce particles pseudotyped with fusion proteins of other viruses, not just coronavirus S. For best results, it is important to monitor cell health and density for transfection as well as for pseudotyped virus infection. Demonstrating this procedure will be Tiffany Tang and Lakshmi graduate students from my laboratory.
To begin, carefully wash HEC293 T cells with 10 milliliters of 37 degrees Celsius pre-warmed DPBS twice. Next, aspirate the supernatant and detach cells with 1 milliliter of 25% trypsin solution that has been pre-warmed at 37 degrees Celsius. Then, incubate the flask of cells at 37 degrees Celsius in 5% carbon dioxide environment for 3 to 5 minutes until cells start detaching. Add 4 milliliters of complete DMEM to deactivate the trypsin solution, and then count cells using a cell counting slide and a light microscope. Dilute cells to 500,000 cells per milliliter with complete DMEM. Next, seed 2 milliliter per well of the cell solution in a 6-well tissue culture plate, and gently move the plate back and forth and side to side to evenly distribute cells. After making sure that cells are evenly distributed, incubate the plate at 37 degrees Celsius in a 5% carbon dioxide.